We're going to continue our look at the immune system and immunity by looking at adaptive or specific immunity. Adaptive immunity is specific to specific invading agents. It's the ability of the body to defend against very specific things. This whole branch of immunity, as opposed to the non-specific, only is activated when certain antigens, when certain pathogens, as it were, are present. So the adaptive immunity that is, for instance, against measles, will do absolutely nothing against influenza. It will be a very similar, the way we fight influenza would be a very similar thing to the way we fight measles, but it won't be the same cells or the same branch of the adaptive immunity. It's like that for every single pathogen. Adaptive immunity is really it's got this idea of specificity in that it's specific for the specific antigen, and it has memory. It remembers when we have seen that pathogen or antigen before and does a better job the next time we see it. So there's this whole idea of memory. Now, one of the fundamental things about that you need to know about adaptive immunity is that it's provoked by characteristics of the pathogen or characteristics of whatever is invading you called antigens. Antigen generates the anti-response. So these are substances that are recognized as foreign, and it provokes the immune response. It makes the immune response happen. Now, in the specific immunity, they are cells that actually respond to the antigen, and their whole idea is to eliminate that antigen. Now, there's the two basic populations of cells. There are T cells that develop in the bone marrow and then become immunocompetent in the thymus, which is why they're T, and there are B cells that come from the bone marrow and then become competent and complete their development in the bone marrow. And really, immunocompetence is about whether that cell, that T cell and B cell, will recognize and react to the antigens. So in order to do that, really what they have to have is a receptor to that antigen and that specific antigen on their cell surface, so that that cell will recognize it. We talk about the two ways that this happens as cell-mediated and antibody-mediated immunity. Now, the T cells all are involved with cell-mediated immunity, and the B cells are involved with antibody-mediated immunity. The basic difference is that the cell-mediated immunity, the cells, the cytotoxic T cells, are actually involved with killing of cells the same way natural killer cells are, and they are the direct actors. In antibody-mediated immunity, the B cells become plasma cells, and then they release antibodies, which are non-living extracellular proteins, as it were, that they do the interaction with the antigen. The cells themselves don't interact with the antigen. So this is a slide about that. The cell-mediated, it's the cytotoxic T cells that directly attack the invading antigens. We use them the same way that we use killer T cells, and they basically attacked any intracellular pathogens that are inside the cells. They will attack cancer cells and foreign tissue implants, but their basic function is to kill tissue cells that have something wrong with them, something specifically wrong with them. The antibody-mediated cells is about the B cells becoming plasma cells, which are basically B cells with more endoplasmic reticulum, so they can make more proteins, and they make antibodies, which is a globular protein involved in the immune system, which is why they're called immunoglobulins. The antibodies don't work inside cells, they only work in the extracellular fluid, in the interstitial fluid. Both these types of specific immunity work together. There's very few things that will be one or the other. And remember that the adaptive immunity takes a little longer to kick in than the non-specific immunity, but the non-specific immunity doesn't stop working when this starts working. So they'll all be working at the same time. So at the same time that this is happening, then macrophages will be eating stuff, and natural killer cells will be killing tissue and liberating into the interstitial space the pathogens. There will be fever, there will be interferons going on, there will be complement being released, there will be antimicrobial proteins and iron binding proteins happening, and all of that inflammation will all be happening while this is going on. Now, one of the first steps when we activate the specific immunity is that in reaction to being activated by an antigen, then what happens is these lymphocytes, the B and T lymphocytes, proliferate. They basically clone themselves, and they make more copies of themselves with that particular specific receptor on it. So that what we end up with is a whole army of cells that are specifically geared to attack or deal with that single pathogen. Both the T and the B cells do it. And so these clones are identical cells to the original cell. So there's two populations of these clones. There's the ones that we use right away, which are called the effector cells, and they get involved with what's happening right now, and then they die when they're no longer needed. And there's another group of these cells that are called memory cells, and they don't get involved with this infection, but what they do is they hang around for years to come. They have very long lifespans, and their job is to be present in case you encounter the same pathogen again. At any given time, if you've never been exposed to the antigen or the pathogen, you have a very small population of cells that are specific to it. Once you've encountered it once, through cloning, you make thousands and thousands and thousands of copies of that cell, and so some of those become memory cells. So instead of the pathogen or the antigen having to randomly meet the specific lymphocyte that works against it, which might take days to happen, by having thousands of memory cells kicking around, then there's much less time delay. And so the second invasion gets responded to much more quickly, and because there's that many more that'll clone, then you reach that critical mass of effector cells much sooner, and usually you don't even get signs of the infection. You usually beat it right away. Now, all of this is provoked by something called antigens. And antigens really have two characteristics. One is they evoke an immune response. We call that immunogenicity, generates the immune response. There are lots of things that will generate the immune response, but that's only half the story. The other half is they have to react with the antibodies that it provokes. And it's one thing, it's kind of like you can piss off a cop, but if you have if he can't arrest you, then it doesn't mean very much. So there has to be this ability for the immune system to react and interact with the antigen for it to be considered an antigen. So what happens is the antigen has got things called antigenic determinants, or epitopes, and they're what triggers the response. Now, it may not be the entire microbe, but it might be. So these antigenic determinants are what create the response. And each antigenic determinant has a specific reaction. It causes a different part of the specific to get involved. Now, some pathogens have many antigenic determinants. So what will happen is they'll get attacked by 3 or 4 or 5 or 6 different branches of the adaptive immunity. Other things have maybe only one antigenic determinant, so the response against it will be much weaker. It's like, what are the characteristics that make it up? Now, an analogy that I use in class, in live class, is for the girls out there, you're attracted to guys that are tall. You're attracted to guys with nice abs. You're attracted to guys that are blonde, that have blue eyes, that are eloquent, they speak well, that are funny, that are well educated, have good jobs, and lots of money. Each one of those things is an antigenic determinant. If the guy has all those antigenic determinants, your reaction to him will be much stronger than if he's a short, dark-haired guy with brown eyes, not very funny, has a lack of education, but has a good job. Or if he's tall, blonde, good-looking, nice abs, and as dumb as a post. The reaction won't be as strong. And it's the exact same thing with this, that the antigen has these determinants, and how strongly you react depends on how many of the determinants are specific for your immune system. There's a billion different epitopes. There's all kinds of different things, and it's all controlled genetically. Now, one of the interesting things about these antigen receptors is that we have receptors for things that we have never ever been exposed to. We are genetically pre-programmed to have these receptors. So you have receptors in your body right now that are programmed to react to smallpox. You have never encountered smallpox. They say smallpox has been eradicated, but our genome has the ability, and we have made lymphocytes against smallpox. We will make no memories cells against them because we've never been exposed. But that's the point, is that you don't have to be exposed in order to recognize and react. Now, every cell in our body, or most cells in our body, have these flags on them, the uniform that they wear. We called them, when I was in school, we used to call them HLAs, human leukocyte antigens, but now they're called major histocompatibility complex antigens. These are the things that tell the immune system that these cells belong here, that they are you. And every single one of us has a unique pattern for their MHCs, with the exception of identical twins, which their histocompatibility complexes are the same, but because genetically they're the same. The MHCs vary from individual to individual. How much different they are depends on how closely you're related. Now, they're used to determine which cells are foreign and which cells are self, which ones do you attack and which ones do you not attack. Now there's two classes of them. The class 1 is built into all the body cells except for red blood cells. Class 2 is in a special type of cell called an antigen presenting cell, and we're going to see the difference between them shortly. But class 1 MHCs are the ones that are just about everywhere. Now, the B cells bind directly to the antigen on the pathogen. They are, because the B cells fight cells in the interstitial fluid, they are looking for antigens that are in the interstitial fluid, so they're whole antigens. T cells, on the other hand, are cells that attack and kill tissue cells, human, like your own tissue cells, that have something wrong with them, something specifically wrong with them. So they only react to fragments of the antigen that gets associated with the MHCs, and it happens to be the MHC 2s that will evoke the response, and antigens in the MHC 1s flag the cells for attack. And so if the pathogen or the antigen is outside the body cell, it's the B cells that will react to it. If the pathogen or antigen is inside a cell, then it is the T cells that will react to it. The term for that is exogenous, present in the fluid outside the body cells. And endogenous are the ones that are inside the body cells. So the exogenous antigens get eaten by macrophages or dendritic cells, or they will activate B cells, and in doing so, they will present a processed part of the antigen to the T cells. And so that antigen becomes part of the MHC2s, class 2, in the plasma membrane, and that's what presents to the T cell and sets it off. The endogenous antigens are the ones that get inside body cells, and their fragments of the antigen get displayed with the MHC 1s, and they basically say, oh, I'm infected, something's wrong with me, come kill me. So what happens is the exogenous antigens get eaten by the phagocyte. The phagocyte digests, kills and digests it, and then as it's making its MHC2 molecules, it takes a fragment of the exogenous, usually the antigenic determinant, processes it, and displays it on the surface. So that's why it's called an antigen presenting cell. It's presenting the antigen with its MHC2s. The endogenous ones is that the antigen ends up in the cell, and as the cell is making its MHC1s, it ends up putting a fragment out and displays it and says, I'm infected, deal with me. I'll end this video now, and then I will pick up and do 2 more videos on cell-mediated immunity and then antibody-mediated immunity separately.